Hello everyone, welcome back to the Genomics Bootcamp. We are continuing our discussion on the report of the World Health Organization's expert committee on the human genome editing. This time we will be discussing the potential use of human genome editing, and this expert committee put together a document which is the framework for governance. Actually, this is one of the two major documents that are the output from this committee's work. And this framework for governance also includes this box 3, which has the title Current Potential and Speculative Human Genome Editing Research. So basically it lists the potential uses of the human genome editing. Also, I would add here that this is a rather long table, but it's focused solely on human use or the human genome editing. It does not include the aspects of the genome editing in other organisms, such as plants, animals, or microorganisms. So there could be many other uses of the genome editing technique as well in the other organisms. Also, well, this is a rather long table, but also it has some notes on the second page. So make sure you check out those notes as well. We will also do so in this video, but well, I just want to underline the existence of these notes that are the integral part of this box 3. Of course, you can download the whole document and check out the data table yourself. I will put the link in the description below. Also, if you have any questions or comments on the topic, I just encourage you to put them into the comments. Also, the usual procedure of hitting the like button, sharing the video with your friends and colleagues, or if you are not subscribed, pushing the subscribe button is much appreciated. So, as the YouTube saying goes, comment, like, and subscribe. But anyway, let's get to this box 3, which is a very neat and well-organized overview of the potential uses. It is color-coded, so the color of the box indicates whether if it's basic or preclinical research in vitro or in animal models in vivo. Then it could be prenatal, in utero, and postnatal somatic genome editing. Then this could be also heritable genome editing. So there are some uses that are plainly heritable, or there is a bunch of techniques that could be either somatic or heritable genome editing. Now I would like to just underline. So these are the two major categories of human editing. One is a treatment, so to treat some existing condition or disorder or disease, and the other one is changing the genome of the individual at the very early stage, for example, an early zygote, and then this change is in every single cell of that organism, and then it's inherited to its offspring. So this is why the somatic genome editing and the heritable genome editing is so clearly distinguished. So the first part is on the basic or preclinical research, and the genome editing could be used to alter genes or their activity in human somatic cells or tissues, including organoids or germline cells, zygotes, early embryos, pluripotent stem cells, embryo models, germ cells, spermatogonial stem cells, gamete precursor cells, or gametes. Or also, it could be used to laboratory animals containing human genes or cells or tissues. And all these alternations of genes or their activity could be used to study human biology and the role of specific genes and processes, for example, development, physiology, and disease. Also to establish models of human genetic disease. Also, these alternations of genes could be used to screen for human genes that are involved in disease or that respond to substances, including potential therapeutic agents and toxic materials. And another use could be to refine techniques of genome editing and test specific reagents for use in somatics and germline human genome editing. The second box is listing possibilities for prenatal, in utero, and postnatal somatic genome editing. So this is the non-heritable genome editing for treatment. And overall, you will see that it lists various opportunities or possibilities to cure existing diseases. Okay, so then back to the document. So the genome editing could be used to treat genetic disorders, to alter genes or their activity ex vivo, e.g. using bone marrow stem cells, or in vivo, e.g. using viral vectors. So basically infecting people with altered viruses and therefore implementing the desired change into the living organism, but this change is not inherited to the offspring of this organism or person. Genome editing then could be used to treat monogenic disorders by either correcting the mutant allele for autosomal recessive, sex chromosome linked, or dominant mutations in the nuclear DNA, or by correcting or eliminating mutant mitochondrial DNA. The second possibility is the deleting the disease-causing variant for dominant mutations, e.g. for the Huntington disease, or making deletions to promote exon skipping, e.g. Duchenne muscular dystrophy. The third option is by boosting the expression of a closely related homologue through inactivating genes encoding repressors or by mutating repressor regulatory elements, e.g. to boost gamma globin gene expression for sickle cell disease or beta thalassemia. And the fourth option is by using so-called safe harbor sites in the genome to integrate a gene whose expression will rescue a loss of function mutation, e.g. one leading to an enzyme deficiency. So we see there is multiple options for treating monogenic disorders, or in general, the alternation of a single genes are considered the most straightforward when it comes to genome editing. Another possible use of genome editing is to boost an immune response against cancer cells, e.g. via chimeric antigen receptor T cells. Well, this is just a short note here, but I think the impact of it could be huge, because if implemented in a way that it works, then this thing here, this could be just basically cure for cancer. So certainly there is a lot of potential for use of genome editing for treatment. Anyway, but coming back to other uses, so the genome editing could be used also to correct somatic mutations in stem cells leading to the disease, e.g. acute myeloid leukemia or chronic lymphocytic leukemia. Also, it could be used to treat polygenic disorders or disorders influenced by both genes and environment, e.g. coronary heart disease, cancer, and autoimmune diseases. I scroll up a little bit here just to remind you that the next section is about heritable genome editing. So this type of genome editing is the one that is being done in presumably the early stage of life, and then it is inherited the offspring, and the offspring can potentially kind of give it over to their offspring. So basically, we are implementing lasting changes into the population. So it is just a smaller box at the moment, but the genome editing could be used to avoid inheritance of genetic disorders. So to correct the mutant allele for monogenic disorders, including autosomal recessive, sex chromosome linked, or dominant mutations in nuclear DNA, or by correcting or eliminating mutant mitochondrial DNA, or to reduce the likelihood of complex multifactorial or polygenic disorders, e.g. coronary heart disease, diabetes, or autoimmune diseases. And the next large part of the possibilities of use are the ones that could be either on the somatic cells, so the non-inherited variant, or the heritable genome editing techniques. So here is a separate category on treatment of infertility. So to alter genes in gonadal supporting cells such as Sertoli or granulosa cells, so that the germ cells can form functional sperm or oocytes. Also, it could be used to correct mutations in germ cells in the testes or ovaries, or in the germline cells used to derive gametes in vitro. Also, the genome editing could be used to promote disease resistance, to alter an allele associated with increased risk of a disease or disorder to the one that is protective. So basically just exchange the problematic allele to a allele that gives you resistance or tolerance to the disease. This could be used to reduce infectious diseases and parasites, for example, by altering human genes encoding pathogen receptors or that allow pathogen replication, so CC5 for HIV, for example. Also, it could be used to reduce cancers due to oncogene activation or tumor suppression mutations, which can involve loss of heterozygosity, e.g. in the BRCA1 gene. And also it could be used to reduce genetic diseases influenced by known genetic risk factors or alleles, for example, the Alzheimer's disease and the ApoE4 versus ApoE2 or 3. Overall, the genome editing also could be used to increase quality of life. That is to alter an allele that may be relatively rare or common to a different common allele. So again, this is about exchanging genes to the ones that provide some better characteristics. For example, to increase tolerance to lactose, gluten, or alcohol for improved diet, to reduce blood cholesterol levels to improve metabolism, and to avoid adverse drug effects or promote better therapy, which we can also look at as so-called reverse pharmacogenomics. And then with these last two boxes, which can be also treatment or heritable changes in the human genome, we are entering almost like a sci-fi territory, because the genome editing could be used to enhance human traits, to alter alleles or to other variants, which may be common or rare, and give extreme characteristics that are present within the family or other human populations. In other words, if we identify a genetic mutation or a gene that provides the desired characteristic, we can ensure that this gene spreads in the population in a quicker way, or we can implement this gene into the desired population. This enhancement can go various directions. We can alter appearance, for example, hair color. We can alter other abilities such as muscle mass or voice characteristics. Also to increase muscle type, height, weight, longevity, or intelligence, to provide resistance to pollutants and other environmental agents such as radiation. Well, obviously, some of these changes are more straightforward and other changes are really speculative. For example, the increase of intelligence that is something that I would be very doubtful about if this could be done at all or in which time horizon. And the next box takes it even further because it talks about adding non-human traits to the human population. So to introduce single or multiple genes not present in any human genome, non-human or synthetic genes to, well, amuse and entertain. So there are some genes that provide fluorescence. This has been shown in mice and fish so that it could be implemented. This is certainly a possibility also in humans. I don't know if the one that we should necessarily follow, but also we can improve sensory systems to ultraviolet or infrared light or to electromagnetic fields to obtain nutritional benefit from parts of plants, plastics, and other materials that humans cannot currently digest, or to increase tolerance to drought, heat, or cold, and to provide resistance to pollutants or other environmental agents such as radiation. Well, certainly these two last boxes are ones that move our fantasy in a big way. And anyway, so there is a specific part on this WHO report on the human enhancement. So there is a specific video on that. So if you're interested in this topic further, I just encourage you to check it out. I will put it also in the description below, and well, you will certainly find it in the channel. So this was the list of potential applications, but here are also the notes that I mentioned that are also very important. And well, these notes say that the examples in box 3 are illustrative and not comprehensive. Also, there are no one-to-one mapping with the content of the governance report or the recommendations has been done in the committee's focus has been on the current and near-term future research involved human genome editing. The examples are also descriptive, not normative. That is, they are simply meant to provide an overview what might be possible with the science. So this says that this is, again, which might be possible. So this is not something that is being done or being planned. The examples do not include methods that might be used, but depending on specific application, these methods could involve repair of double-stranded DNA breaks, non-homologous end joining, and homology-directed repair, NICs or single-strand cuts, base editing, and prime editing, and RNA or epigenetic editing, where the last two are unlikely to be heritable. Also, it is underlined that clear differences in the complexity of the science and ethical considerations will exist depending on whether the attempt is to modify one or several genes. With the current genome editing techniques, altering multiple genes simultaneously would greatly increase the likelihood of, for example, incorrect on-target and off-target events and chromosome rearrangement, such that the risk outweighed any potential benefits. Also, in the future, some applications of human genome editing might involve the use of techniques to increase rates of inheritance, for example, using gene drive technologies to allow humans to cope with extreme climate change. So here, the most important or interesting part is the gene drive technologies, which are, I do not go into detail right now, but it certainly deserves a closer look because these gene drive technologies are very interesting and could hold a great promise, but also a great danger, depending on how they will be applied. And following up with these notes, they also mention that there are also potential dual-use applications, for example, human genome editing to give resistance to chemical pollutants or to radiation for space travel, could also have military applications with respect to resistance to chemical or nuclear weapons. Yeah, so this point will come back again in the other video with the human enhancements or the use of genome editing for human enhancement. On multiple parts in this report, the military use is mentioned. So I think military applications of the human genome editing are foreseen. And also this last point, that it is important to highlight that the committee does not endorse or even suggest that any researcher works towards these potential uses. Rather, the committee advocates the introduction and implementation of national and transnational governance mechanisms that can effectively review and assess not just scientific and clinical evidence, but also relevant ethical and societal views and values. So thank you very much for your time for this video. If you're interested in a similar content, be sure to like the video, send it to your colleagues. And if you have, of course, any questions, comments, or I would be also really interested in your opinion in the subject, make sure that you write those in the comments below. So again, thank you for your time and have a very nice day.